Hello again. Here I am now to another lecture about the cell properties and behaviors. This time I'm going to talk about cell growth and cell division. Uh, I forgot to tell the first time that this lecture is being live streamed, recorded, and distributed by YouTube. Okay. Uh, okay. In these lectures, I will present you these new tools that CompuCell offers to you, that are tools that make your cell growth and eventually divide through a mitotic or mitosis event. Here's the outline. I will present, as usual, a brief overview about the motivation to why to include cell growth and division in the as tools in the simulation, in the CompuCell software. And then I will present you three exercises implementing this cell growth and division with the mitosis steppable, which we are going to introduce today. As a bonus, I think I hope I can introduce you a way to open graphic windows in your player to see the evolution of some quantities during the simulation. Okay. Uh, let's start then with this small overview. This slide we already saw this morning with James. It tells you about um, uh, states that the cell can achieve, can be during his life. And those states are basically proliferation, survival, differentiation, and death. Okay? There are many factors, even internals or externals, that may contribute for the cell to be in one of these states. Okay? For example, growth factors promote cell proliferation in in an organism. On the other hand, there is some inhibition of proliferation that occurs because of the environment where the cells are too crowded. I mean, when the cells start to contact each other, this contact between them produces or inhibits the proliferation of them. So there is a bunch of other factors that can promote those four outcomes of the cell can be, which leads us to the concept of a cell state for a cell. We can say now that a cell is in a state of proliferation, a state of survival, or anything else. Well, when a cell decides to proliferate, I mean the decision is made through some external influence, for example, it enters which we call today as the cell cycle. The cell cycle is a set of events that occurs between one division and the next one. I mean, the cell divides and then it starts a new cycle. During this cycle, it passes through a series of the events, and these events are identified in this circle here by regions where the cells stay during a certain amount of time. And the regions are G1, S, G2, and M. the G1 to G2 is called the interphase events, and is the time of the cell where they start increasing in size, production of RNA to synthesize proteins. Then it enters in the S phase to synthesize DNA, goes to G2 to production new proteins and prepare the cell for division. That eventually occurs in the M phase, which is the mitotic phase or metaphase. In particular, this metaphase is compounded by four subphases, let's say that way. Okay. Um, the interesting is that each time that a cell uh, ends ends a phase and it's about to enter in another one, for example, from G1 to S, the cell um, fires a set of internal mechanism that we call checkpoints, and those checkpoints test the integrity of the cell. I mean, it tests if the cell during the previous phase that is finishing, everything was well done. If ever see something fails, one of the checkpoint fails between GAS, S, and G2 and M, the cells stop to the cycle, entering a rested phase, and eventually die by apoptosis. Okay? So we are not going to now to simulate or propose some model to build the entire cell cycle in details. This is a very big task. We are now going to implement some those mechanisms that allowed this uh, proliferation and division, eventually the mitosis event, which we call cell growth and cell division. You can see here a set of cells in a cycle that ends by the division. You can clearly see the nucleus being divided and the DNA being split in two, and the cell finally divided into different cells, separated cells of the same type. Yeah? However, to understand or to be able to implement cell growth and cell division, we have to ask what could affect, for example, for cell growth, the growth rate of the cells. For example, we can take into account the space availability for the cells to grow, since the contact could inhibit the growth. If the environment has enough nutrients for the cells to grow, how fast the cells can convert the nutrients into mass of them, and how efficient is this conversion? Are there growth factors in the medium? Are there contact with the cells with the substrate? The substrate can be ECM, could be in fluid, something different in where the cells can interact. What is the internal state of the cell? And the important thing, are there toxins in the medium that could damage the cell? So there is a bunch of things that we could consider to produce the to implement the cell growth. But we are going to be simple here. We are going to forget most of these um, these items here, and to establish some very simple rules for the growing. Let's say the following. We know when we define the cell we define a target volume for the cell. So we are going this way here to implement a cell growth by summing to the target volume a constant value at each time step or if some amount of time steps during the simulation. Then we can change the volume of the cells, change its target volume. Okay. The thing is, differently from the previous lectures where we define the target volume and the lambda volume in the XML file. In this case, where we have to change these parameters, we are not to do that in XML file. We cannot do that there. The XML file, once is loaded with some parameters, that parameters stays there forever. You cannot change. So you have to now use another way to do that. And this is done in the steppable file. Remember, the steppable file is the place where we uh, implement user-defined calculations, functions, graphs, anything you want to do the simulation calculate that is not in the plugins in the original steppables. So what we're going to do is in the steppable has that initial part, the start function. In that start function, we are going to define the initial value for the target volume and for the lambda volume for each cell in our simulation. So supposing that we start with many cells, okay? We do a loop for all the cells, and for each cells, we attribute the target volume and the some lambda volume. This is a different plugin because this plugin allows you to attribute those quantities by cell type. If you want to do two cell types, for example, and those cell types have different lambda volumes, for example, you can define for each cell type here in the step. Okay? The change in the target value is made in the step function of this steppable. Remember, the start function only works once at the beginning of the simulation. The step function runs each Monte Carlo step or every amount of Monte Carlo step you define. So, for example, here we are saying that we want the target volume to increase 0.1 voxel per Monte Carlo step. It means that it takes to 100 Monte Carlo step to a cell increase 1 pixel, 1 vox. This is done in the steppable file. Huh? So let's see how to implement this in this initial exercise. Here, let's start to edit with a new project, this one. Call it cell growth if you want. Again, there is the cell growth simulation ready to run in the student materials demo codes folder. If you want to download it, be my guest. So if you start a new simulation, let's go on. You have the grid. You size the grid 100 by 100. But now, instead the rectangular slab, we use block. Go to the next window and create only one cell, only one cell type. Name is cell, for example. And now we include in the cell behaviors and properties the following plugins: contact, volume local flex. See, remember in the previous lectures we include volume local. Now it's volume local flex, surface local flex, and center of mass. And add the steppable growth at the left panel in the window. Now close the simulation, finish the simulation, open with it, and let's go to edit the blob initializer in the XML file. The blob initializer is usually at the end of the XML file. Okay? Put in then radius equal to 5 and width equals to 5. The difference between local flex and local is that local flex allows you to define the parameters per cell type. And it is defined in the steppable. The single local volume or surface, is defined for all the cells, and it is defined in the XML file. The same for surface local flex plugin. Okay, after you edit the blob initializer, put in radius 5 and width 5, go to steppable function. Steppable file, sorry. The steppable file has a class called constraint initialize, which is there to initialize the steppable, the plugins we just loaded in the XML file. Okay, so let's go to the start function. You have to write now, type in cell target volume equals to, well, sorry, let me go in another, I have to go, forgot to put the image here. I would, for a few minutes to see here. In the steppable, you have the start function that comes from nothing in principle. Then you have to include this loop to attribute to the cell what are its initial target volumes, lambda volumes, target surface, and lambda source. How do you include this line here, the loop? You can use the drop-down menu here in CC3D Python tab, visit cells, all cells. In clicking here, that line appears there. Then you have to fill the four following lines, targets and lambdas. Okay? This is what we said in this first line, step 3 of the simulation. Okay. The next step is to edit the growth steppable that you have also loaded in the wizard. There it is. The growth steppable has here a line in the start function where you, sorry, here, the step function, you have to include again a line where you make the loop for the cells, this line here. You include that line. That line will give you the growth rate for the cells. Here I'm saying that the cells increase a quarter of a voxel per Monte Carlo step. Yeah? Let me follow. Everybody could do that? Remember, we are in the very beginning. You don't have anything else besides this line you have to adjust here. Adjust the cell growth rate to 25 voxels per 500. 500 Monte Carlo step. I put there 100. Okay, then we change later. Next, now that's a new thing. We are going to implement a graphical window in our simulation to follow the growth of the cell in a curve. Okay? How do you do that? Again, in the start function here, you come to the CC3D Python tab and go to scientific plots. In the scientific plots, you go to the setup part. Clicking here, in a snippet with the plot window will appear. It comes with some definitions as default, but we can edit it. And how can we edit that? This way. Yeah? We have to edit the title, put the title we want, the title for the x-axis, Monte Carlo step, the title for the y-axis, volume, the x and y scale. This is an interesting thing because you can use the logarithmic scale if you want. But for now, let it as linear in both scales. Linear, you have a Cartesian coordinate system with linear scale. And we don't want to show any grid in the graph. Then, in this line here that comes with the snippet, you give to this window a name. I mean, at this window named vol, you are going to plot with lines in color red of size 5. I mean, the width of the line that is going to use to plot the volume at each Monte Carlo step. Okay? I already have the following one here, but I will show you this later. Now you can run the simulation. Oops. Let's see the simulation. Where is that? Here. I think I'm going to pass this one and have very quickly these steps. Here is where you should change. Change. Steppable changes here. This is how the steppable looks like when you start the simulation. There is a line for run over cell ID in the step function, but in the start function, there is nothing. Then you have to include here the initial values for the target and the lambda. Okay? And in the growth, you add this line. You adjust this line. I put 500 here by mistake. It's 100. So interact, configure the graph here, add, and the last thing you have to do is to include in the step function of the growth steppable a line to plot the value for the target volume at that Monte Carlo step. So you're going to plot in the window that I call vol. Let me put where is pointer here. See, in the window vol, which is here, with lines, we're going to put coordinate x is MCS, coordinate y is the cell volume. This line is also put in your code using this CC3D Python scientific plots item 2, add data point. So the line comes as a snippet, and you just have to to write the right values for window and what are the set of points you want to plot. Okay? Next. Not good. If you put the simulation to run now, you should see this thing. Please try it. If everything goes right, the cells grows forever. Slowly, but forever. Eventually, the cell will occupy all the space because its target volume is increasing with time. So there is no limit for the growth. Everybody could see that? Check the code then. No, not surface constraint now. Well, maybe you could put it to 0 if you want at the beginning, because we are only looking at the growth together with the graph to see if that the cell grows. Let me go to the other. Okay, now the next step is to include the cell target and the cell lambda volume in the start function. It's funny. Let's see what happened there in the XML. See, the XML doesn't have anything, only the load, the plugins that we have loaded, volume and surface and center of mass, cell type, and the blob initializer. In the blob initializer, we have this radius 5 and width 5, and cell type equals to cell. Yes,
Oh,
the target growth is The target volume is always growing, so the cell keeps growing. Let's look at the plot window. Here's the plot window. Title, axis, volume, scale, grid. And I'm going to run my code here. My code runs fine.
Gilberto,
Yeah.
one thing you might want to do, because people are having some issues in debugging, is introduce a Python error and show them how to find the line with the error in it in the player window
Oh,
to help
okay.
people debug.
Okay. When the CompuCell crashes in the, well, we have two windows actually. One for to edit, and when you open the player, if you open the player from inside the to edit, the information about the running will appear here in the same to edit windows. So if something happens in the player, like an error, this error should appear here are in the in this window as a line. Error such that. It tells you the line where the usually tells you where the line where the error is occurring. I am in the version 4.6 here. This version is 4.6. Okay, you can see, try to find out what is going on, what is the error if you have something typed wrong in this window here. If you don't have CompuCell, the player open from inside, from an extra window, then the error line will appear in the command window that CompuCell opens when it starts. Oh, what's going on? Yes, I can. In a second. Here. Since I didn't have any error, there is no line error here. See, I will start CompuCell from here to see. Oh, see, opening player. Here, all the instructions about the player running in this window. And there is no error. The simulation is running fine. See? We have the volume and we have the surface. When I stop the simulation here, the simulator here in this window also, this, the simulation stops and come back to information about the twist. Could you identify the error? Let's see what is this message. Ah, it's a Mac? No. Can anyone help Mark with this error?
Pedro, are you here?
Seems no.
Oh,
Oi, Pedro.
Pedro,
am a little bit here.
could you possibly go into a breakout room with the people who are having trouble and work with them a little bit?
Okay, I'll stay in room 1.
I can't, because I own the meeting, I can't go to a breakout room, so I can't do that.
Oh,
So people who are having crashes, repeated crashes, please join Pedro in breakout room 1. Is that clear for everybody?
so now, Shane?
Gilberto, why don't you focus on
Okay.
exercises for the people who are
Yes, I
making.
was trying to understand the question from Clara. Okay,
So
since
Clara, why don't you put a picture in the in the uh, chat of what you're getting?
okay, I will go now. The next is to include the surface plugin, to put the surface and plugin in motion, saying we have to include this surface, target surface and lambda surfaces. This is done again in the start function of the constraint initialized step. Okay, and to see the evolution of the surface, of the cell surface, we add this next window in the same graphic plot we have. So we can have a graph. In this graph, you can have many plots. Each plot has to be defined with a name. For example, here I have this example. We are going to plot in the same window volume and surface of the cell. So we have two plots. So we have to add two windows to that graph. These windows, one is called vol, was the first one, and the second I called sur, for the surface. Again, I define lines as the style to plot. I change the color to blue, and the size is the same, 5. The same thing I have to do in the step function, because now I have to add the plot to that window that I just created, the sur window. And the syntax is the same. Self plot window, blah, 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 blah. Sur, now I'm plotting x coordinate, MCS, and y coordinate, the surface. So I'm plotting surface against time, or MCS. Yes, the radius is 5, and width is also 5. Now we can run the simulation with those two plugins considered. Yeah? When I do that, we have this kind of behavior. The cell evolving like a square. That's what you should see. Because although the target volume of the cell is growing, the surface target is kept constant. So the surface target prevents the cell to grow. I mean, prevents the cell to grow in a rounded form, if you want, because it's trying to keep the surface of 20 for the cell. Then you have to fix that. Oh, remember what we have used for the previous relationship between surface and volume? We are going to use here this one. We are saying that the cell is a circle, and the surface is the perimeter of that circle. So when I actually the cell is a square, and the surface is the perimeter is a circle. So I calculated the size of the side of the square, and I'm going to say that is the radius of the new cell. And so the perimeter is 2 pi times the radius. And I include that here. See, to implement this equation here to make the variation of the cell's target surface, this is done in the growth steppable together with cell target volume growth. But since we have the pi here, I have to define pi at the beginning. So pi is 3.1415, blah, 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 blah. And then I write that equation here. 2 times pi times square root of target volume. And add a line for to plot the blue line here. So you have the evolution is more circumferential now because the surface, the boundary of the cell, is adapting to the volume of the cell at the same time. So both grows simultaneously. And the graph you have is this one. A linear growing here because the growth is linear. This is a constant factor here. And a power law here because the surface grows as a square root of the volume. So it's a curve here. Yeah? Could you see that, people? Take a minute to implement those lines. Here is the simulation. It grows indefinitely. There is a bug with the axis here in this window. The axis doesn't grow with the window. Oh, monarch is okay. Mark is okay. It seems that people is getting the right images. Okay, let me go on then. Now you know how to make the cell grow. We have to adjust both volume targets and surface targets. Now we have start to thinking about how to make the cell to divide. Here we have this small movie showing a specific cell dividing, separating its DNA, separating themselves, and creating two new cells. What triggers cell division? I mean, what? To talk about mitosis then, which is a very complex process with many sub-steps that you just could see. And we mentioned at the beginning during the cell type. Remember, the metaphase phase has 4 different stages to achieve until the division. And cells that destroy nucleus, contract genes, pull them apart, divide and recreate the nucleus. This is a very huge task. Okay? But we can start with a simple model. We are not going to model those metaphase stage. Instead, the simple model that we're going to say, implement is say that when the cell grows and reaches the double of the size, the initial size, it divides. It's a simple condition. So we have to keep track of the cell volume at each Monte Carlo step. When it reaches the double of the initial value, we will implement the mitosis process. So when we have the cell division in general, we have one cell becoming 2. So you need to create a new cell. For the sake of terminology, let's say that we have at the beginning one parent cell, and in biology we say that we have 2 daughter or child cells. And those cells have to implement the division. After the division, or even to divide, we have to choose make some decisions. For example, which of them will be the parent? Which parts of the parent will become daughter? I mean, is the division equally? I mean, in the half of the cell, the initial cell, what is the plane of the division? Could it be axial? Could it be at any orientation? Could it medial? No? And after division, the resultant cells, do they have the same properties, behaviors as the initial cell? Or after division, the parent cell die or continues his entering a new cycle? Remember, in reality, a cell, a biological cell has a certain limit for the number of divisions that it can make. Okay, so unless it is a cancer cells, which will divide forever, a regular cell has a fixed number or something fixed number of divisions during its lifetime. So that's are things that we have to think about before we start division. Yeah? Okay, let's say this CompuCell has this tool inside his engine. It is a special steppable called mitosis step. This mitosis steppable is loaded at the beginning together with cell properties and behaviors inside the wizard. And it comes with basic operations to perform the cell division and define the attributes for the resulting cells after division. Uh, um, those things like final target volume and surface must be adjusted inside in the configuration of the mitosis step at the beginning of the simulation. Otherwise, you cannot do this later. So, as I said before, we have to, the cell division is triggered by a particular event. Okay, so what event will trigger this mitosis in our case? Well, the simplest one that I have talked already. The, the size of the cell. Here's the structure. Let's have a look in the structure of the mitosis steppable. Here is the steppable. A has an initial function here, and in the step function, it has three different stages, steps. First, there is a line where we can create a list for the cells that are going to divide. I mean, that cells that reach the double of the size, for example. Then it starts with 0 cells, of course. Then we run a loop over the cell volumes to see if any of the cells has reached the volume above 50. If it has a volume above 50, append this cell to that list. Okay? Cells to divide, append cell. Next, we have to decide what kind of orientation we want to divide the cell. We have several options here. Vector beta, you define an axis based in coordinates of the cell, or along the major axis of the cell. And for that, you're going to need to use the center of mass plugin to do that, or minor axis of the cell. We are choosing here to randomly divide the cell in a random orientation. Okay, so you have a loop in that list. And so if there is cells in that list, we are going to say those cells will divide randomly. Okay, so after that, we have to update the attributes of the resulting cells. This is made in this function here, define attributes. So what happened here? We are going to say that, remember I said that we have to define cell target and cell volume by hand here. So I'm saying that after division, the target volume of the parent cell, which is the cell which was divided into, has half of the volume it has when dividing. So it comes back to 25 here in our case. And in this line, it's an interesting line, a very important line, self-clone parent to child. What this line does? It copy from the parent cell all the attributes that the parent cell has and put in the child cell. So the child cell is exactly equal to the parent one. It's a duplicate. The only thing that is different is, of course, the cell ID of the child. When you have the division, the child cell has a new ID, a cell ID. To be able to, we cannot have two cells with the same ID in the simulation. It would crash. Okay. So in this simulation, when we have only one type of cell, the cell splits into equal cells. One thing we could do, if we start the simulation with two kinds of cells, we can choose how the types of the resultant cells will be. For example, in this frame here, I say, I have two cell types in the simulation. If self-parent cell type equals to 1, then the child will be type 2, will change type. Else means if self-parent self-type equals to 2, the child will be 1. I mean, the child's cell swap types with the parent. They will never be the same. But the parent will be always the same type. The parent doesn't change. The child changes. This is a way to create separate types of cells in your to create the different types of cells in your division. Yeah? Okay, let's go there. So the condition then you have chosen is to double the volume. This is put inside the mitosis steppable in this line here. This line is already there. You have just to write the condition. Yeah? You have this list, iterates of the cells, double the cell, test, append the list. This is the view of the mitosis steppable that divide the cell. Yeah? So let's perform the division. Now I have the divided. Now we have the dividing cells. You divide by random rotation, reduce the volume of the parent by 2, I mean, divide by 2, and clone the parent. Okay, this is what I'm going to do a little bit faster here. Well, one thing you have to, this is a general warning that I remember to comment with you. When you create variables inside one steppable, you cannot readily use it in another steppable. That has to be made with some special function that you call self-steppable shared vars, a global dictionary called this way. Then you can create variables in one steppable and use in another one. Otherwise, you have to define it again or store in a file and then read it in another place. It's a kind of boring, but that is like Python works. Okay? But now, before to go to the exercise, let's get used with the mitosis step terminology. Again, self-parent cell is the cell which are going to be divided. When you start, you have only one parent cell, which is a single object. Self-child cell, it's a new cell. It's a new object that has a new ID. And the attributes is up to you to decide. You can clone the cell if it's the same, and if you can put some other attributes. So after mitosis, you have two cells, parent and child. We keep these names after the mitosis because biologically you have two daughters. We don't have a parent anymore after a division. But here, for sake of identification, who is the cell who originates the second one, we keep the name parent for that, for it. Okay? Going home, see, this is what I have there. Okay, now I think we are ready to make the exercise. Let's start this one. Here's the whole exercise now. I think I'm putting it here, but I'm going step by step in the next slide. Okay, let's start from the scratch. Starting to edit and create a new project. Okay, let's go there. Every single step that 1, 2, 3, it's gonna be written in the next slides. Okay? So you can follow what we are going to do. Here, you start to edit, create a new project, blah, 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 blah. The first part we already know. Now we are going to introduce two cell types, STEAM, ST, and TD for terminally differentiated. It means not a STEAM cell. Yeah? And the plugins are contact, volume flex, center of mass, and the steppables growth, and the new one, mitosis here in blue. So next, after we don't have nothing to do in the fields, ignore the fields, finish the simulation, go to the blob initializer again, adjust the region to be of radius 5 and with width 5, and create only one stem cells, ST cells, in the center of the region, the grid. Go to the constraint initialize steppable, and in the start function, put the target volume equals to 25, the lambda volume equals to 5, see, in the start function. And in the growth rate, you put the growth ratio equals to 25/100. Watch, be careful to write the numbers in the growth rate as real numbers. See? Because you are doing here a division. If 25 is an integer, divided for a bigger one, you have always a 0. You have to say that they are real numbers to get the decimal values. So now in this, well, let's adjust the steppable, the mitosis steppable by using the swap between cells here. After choosing the same thing as we have before, cell volume is the condition for dividing is double of the volume. The division occurs in a random orientation, and the parent cell acquires the initial volume of 25, and we clone the parents with the child with the parent. But see here we are swapping types. Say that if the parent is 1, the child is 2, and if the parent is 2, the child is 1. Now you can finish, open instrument simulation, the player. Have a few minutes to edit these things, to see if everything is okay. This is what you should see after a few Monte Carlo steps. We start with initial cell, here, square, and then this should be round here. I think I got a wrong picture. Anyway, the cells start to grow and divide to reach some configuration like that. The cells grow at the same rate because we have said that in the configuration of the simulation. Now let's try something else. Let's change a little bit the trigger for the division. So go back to the mitosis steppable and decrease the trigger from 50 to 25. Oh, sorry, for 30. From 50 to 30. And run the simulation again. You should see that the cells are smaller because now after the division, the cells acquire the size of 15 because it's the half of 30, remember? But it grows much faster because the growth, the trigger limit is smaller. One thing more in this same example. Let's prevent that the TD cells divide. As usual, only the stem cells divide forever. So we are going to edit again the mitosis steppable and change the line for self in self-cell list by the line for self in self-cell list by type self-ST. This line can be included in your code using again, I'm going to suggest to show you here from CompuCell3D Python, visit, or instead, all cells of a given type. That will put that line there. Then you have only to say what type you want to make the loop. Yeah? So this way, only the cell types ST will be allowed to divide. When you run the simulations, you have this. Do it and see if you get this image here. Yes, that's true. See, in these pictures, I didn't go so far. I didn't finish the simulation, but see, we have here the yellow cell, which is a stem cell, a yellow one, is the only one to divide. It divides in two. The child cell, it's a TD cell, okay? Then the ST cell continues to grow and continues to divide, and so on, and so on. After each division, the ST cell, which is a parity, remains, and we have a new TD cells. So the number of TD cells increase. And besides of that, since we are not making any control in the surface or in the volume of the TD cells, they keep growing forever. And that's why we have this big blob of bigger, bigger TD cells growing forever. And if you let the simulation run at the end, they will fill up all the space. Okay? So this is a way to keep only one stem cells. See, this is what cancer do. One single cells making a mess. Okay, let's go on. Okay, so what we just look at, if you let the simulation grow, the space will be filled, totally filled. And when that happens, of course, there is no more space for the cells, then they stop to divide because they cannot grow anymore. If they don't grow anymore, they can reach the, 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 the trigger. And even if they get the trigger, they don't have medium to grow over it. They cannot grow, a cell cannot grow cell over cell because both cells have the same properties. If one of the cells have different target volumes or different properties somehow, one cell could eat another one, growing over it, eventually killing the other. But in this model, all the cells have the same properties. Then they will grow and will fill the space like that. Uh, since the simulation continues, it means that the target volume of the cells continues to grow, despite the cells cannot divide them anymore. So that creates what we call a pressure between cells. The cells become under pressure because they want to grow, but they have no space to grow. Okay? This pressure as a physical quantity can be obtained from the energy term. Let's do a little bit of physics here. Okay? That's it. So, in physics, force is a negative of a derivative of energy, potential energy. Okay? And pressure is the ratio between applied force over the area where the force is applied. So combining just these two expressions here, substituting in here the expression for force, we have this equation here. And in the denominator, we have area times space, which is dimensionally of volume. So pressure is the negative of derivative of energy in relationship to volume where the energy is contained. Okay? Physics. P equals to that. So we have the expression to calculate the volume. Is that one? The derivative of volume energy respect to the volume of the cell. Well, the volume of the cell is that. We know that already since the previous lecture. Is the lambda times the displacement square. So what we have to do now is to derive this expression in respect to the volume, like this, and get this expression. Note that you have this minus signal here. And when we make the derivative, this minus enter into parentheses, change the position of VTG, the target and volume here. Okay? So pressure is measuring a quantity measured from how much the volume departs from the target one linearly. Here is quadratic, is squared. Here is linear. See the difference? Yeah? So you have this. We have a way to calculate a expression to calculate pressure in our simulation because we have the cell volume and the cell target volume and the lambda volume, which comes from here. We are able to calculate pressure in the simulation. Let's go there and calculate. How to use pressure to control cell growth. I mean, we can now use this pressure, the concept of pressure, and the value of the pressure in the system to inhibit, for example, the proliferation of cells, or to as a surrogate for cell-cell contact area. I mean, and to stop the volume of the cell and the surface of cell to increase indefinitely. Okay? Let's see how to do that. Remember, this is what we have for control of the growth so far. The growth is increasing by a constant factor G, which has 25 under 100. Remember that? Okay. Now we are going to substitute this constant value for a function of the real pressure in this simulation. A function of pressure Fp. Say, we are going to increase the volume target by the value of this function of pressure. Okay, now it's not a linear dependence anymore. It can be any shape because F, the function of pressure, can be anything. For example, here I showed you three different shapes that you can use for FP to control the growth rate. You can have a linear decrease in here. What's that? A Hill function? No, a Michaelis-Menten function here. And Hill function here, for example. Okay, so let's use one of them. Of course, we are going to use the simplest one, the first one, the linear one, here. So with the same simulation we have before, we are going to include now the influence of the pressure. Okay? Here is the steps, but I'm gonna again put slide by slide, doing each one of the steps 5, 6, and 7, because 1 to 4 is the same as we have before, the same simulation before. You can edit the old simulation if you want and go from there. But anyway, the simulation, the total new simulation is in the file E232 cell division in the student materials. So now we have, we will edit the old exercise. If you want to create a new one called say pressure. Okay, here we go to step 5 and modify the start function of the growth steppable to include a line with a graph plot to plot the average volume of the cells. Because now you have a bunch of cells. You have to keep track of the average of the cells to calculate an average pressure. Okay, but let's start with average plot. How do you calculate the average plot? Okay, if you want to see the average plot, we include this line here, this plot window to plot, and this window we call AV vol. This will be plot with dots, red dots, size 5. And in the step function, we are going to edit it to calculate the average volume. This is done in this frame here. Look, I create at the beginning a variable. You put it equal to 0. This variable is the average volume. A vol is equal to 0. Then I measured how many cells I have in my cell list. This is what this line do. Length of cell list gives you how many elements do you have in the list. Okay? Then I go over the cell list. And I put in the increment the AVOL variable by the target volume of the cell that is in that list divided by L. Did you get this part? Do you understand that? What I'm doing here in this division? I could sum everything. I could sum everything, and after the loop ends, I divide A vol by L. It's an average. Instead, I am dividing the parcels of the sum by L. That's what I'm doing. The final result is the real average volume of the cells in my simulation. Questions? Okay, moving on. So once I have the value of the average volume, I can plot it in that window AV vol here. See? Add the data point as we did before. In the window AV vol, we plot the axis MCS, x-axis MCS, and y-axis AVol. And we update the cell target volume. We keep increasing the cell volume. See, this is made at each Monte Carlo step. At each Monte Carlo step, before I increment the volume, because the cells have already gained its size, adapted its volume to the target volume, which was adjusted in the previous Monte Carlo step, and then I can make real measurements about the volumes, the target volume. So I do this now. Okay? Then I update the target volumes, and the simulations go on. So see this line, you have to, this frame here, this head box, you have to type in. There is no snippet for this. Of course, there are, to ones who knows Python, you could use NumPy to make this statistics on arrays. So you could use NumPy average, NumPy standard deviation, something. But we don't. We can do this very simple way here. This very simple way here. Oh yes, I think I made a copy paste wrong here. Is the cell volume I should increase that. Let's see what I have. No, remember, because some of the cells after divide, no, it's right. It's not the same target because the cells divide at the different time steps. So the target volumes for the cells may be different. Okay, Kumar? Because of the stochasticity, they don't acquire the same target volume at the same time. They grow differently. Okay, let's go on. Run the simulation. Finish the simulation, run the simulation. This is what we are going to see. Now we are not imposing any restriction about the TD division. Both cells can divide. So we will start with the cells. The cells grows, and fill up all the space. Look, in the plot window, you see the average volume growing. Here they have a division and start growing again, division, start growing again, division, so on, until the space, until the space divide. Uh, sorry, until the space is filled. When this occurs, the cells stop to divide, and the target continues grow indefinitely. The simulations freeze, of course, but the target volumes is still growing because we didn't stop it to grow. Would everybody see that? Remember, AVOL, did you write AV capitals? Remember, you have to be consistent with the, let's see, what is that? the division. No. It's not this one, sorry. I have, see here, I will back one slide here. Here, when you divide, when you create the A vol here, you cannot mistype, it puts a different name here. Did you fix it? Okay. So, this is what happened. Somebody help Troy, please. Okay, I will move on here because time is ending. Okay, we have the frozen system, but the pressure is increasing all the time. So let's keep track on the pressure, the average pressure of the cells. And to do that, we will include now a dictionary, a cell dictionary to keep track of the cell pressure. This is done on the start function of the constraint initializer stepper. Put an entry to the dictionary called pressure, put equals to 0 here to begin. So all the cells in your initial list has pressure 0. Okay, Maria has a strange graph, but I think the graph is right, but the graph is being filled underneath the lines, something like that. Okay, continue. We add this line and a second plot window in the growth steppable, which now it's a really new graph window because the quantities are very different. The average volume, average pressure are much different. So we need a new plot window to track it. And this is the way. We create a new plot window, give it a name, titles, and create a plot named AV press with lines, blue lines, size 5. Okay? And this is what we have to do for initialize quantities. Now we have to calculate the quantity. Which quantity? The average pressure. So to calculate, to be able to plot the average pressure, we have to calculate. So we calculate it in the very same way we calculated the average volume. We define the 0 value for the average at the beginning, and we add inside the loop, the previous loop where we were calculating the target, average target, we add this line. We calculate the actual pressure for the cell in the list based in that formula we just calculated in the physics part of the slide. 2 times cell lambda volume times cell target volume minus cell dot volume. And we add this value to the average value divided by L in parcels, like we did in the average volume. At the end of the loop, we will have the average pressure calculated. And we can plot in the second graph window, Win2. See, I forgot to tell that. When we include a second graph window, we need to name it differently. For example, the first one is self-plot win. The second one, if you use the snippet, will come with the same self-plot win. But then you have to change it win to Win2 because it's a second win. Otherwise, you write over that one. And every time you use self-plot win, like here, self-plot win add plot, again, you have to change win from nothing to Win2. And when you add the data point at the end, you have to add to the second plot window, self-plot Win2, add data point, Windows, plot window AV press, MCS, APRE, which is the target, the average value. That was too hard. Please take a minute to edit the code before we run it. No, that's if you were able to complete the edition and run the simulation, this is what you should see. Now we can see that since the target average is increasing indefinitely, the pressure also is increasing indefinitely. Now, how do you control that? Everybody could see that? Okay, then let's control it. Let's use now the simplest function to replace that constant growth for the target volume for this kind of function here. Okay, so this function imposes you a penalty if you decrease or change the pressure too much far from a preferential value for the pressure you want for your simulation, for yourselves. Okay? So how to implement that? We go to growth steppable and add to the line 25 over 100 this function, which gives you the maximum between 0 and the difference between P reference and actual pressure, the cell pressure. So if the pressure is above of cell preference, this number is negative. So you get 0 here multiplying the growth, and the cell will not grow. If the cell pressure is below this reference value, the function will give you the difference, which is a positive value that multiplies the old growth rate, and the cell will grow. Did you get it? Let's see. This is after edited this line in your code and put it to run again. That's is what we will see. After filled all the space, the target volume stops to grow and the average value reaches a constant value here, and the average pressure also reaches a plateau here. Because since the targets are not growing anymore, the pressure doesn't increase anymore. Okay, I hope you have good, very good. Okay, to end, as an exercise that you can do tonight, for example, you can replace that function, the control function for those two examples here, and see what happens with your simulation. You have to adjust, for example, what is the value of P reference and how much is the power of your U function, and that's it. I hope you are able to do that. I finish here. I hope you sorry by the initial a little bit confused in the presentation, but at the end, I think we could manage to get the results I want you to see. Thank you very much.
Okay, well, thank you, Gilberto, and thank you everybody for your attention to our last session today. We can stick around for a little while for Q&A if people have questions about today's content or about hackathon prep or anything like that. And we will see you back here tomorrow at 10 a.m. for day 3.